Hello dear student, welcome to Cell Biology series. This is episode 4, part 1. I am Dr. Kalpna Rakholiya. Today we are going to discuss about overview of cell organelles. So what we will cover in this lecture? What are the cell organelles? Different types of organelles. These are the different organelles that we are going to cover. Then structure and function of these organelles. So let's start about first lecture, what are cell organelles? So cell organelles are specialized structure that is present inside the cell. So cell perform very specific function and that necessary for our survival. So I will give you one example. Just as our body organ performs specific function to keep us alive. For example, our heart is going to beat like that. Cell also perform the task, as well as stomach do the digestion, and lung keep us alive for by respiratory system. So these are the similarly cell also do the function. Now this image shows the major cell organelles and this picture shows the 3D image. So you can see here, this is the nucleus. Here is mitochondria. This is the endoplasmic reticulum. You can see that here is a no ribosome attached on the endoplasmic reticulum. So these are the smooth endoplasmic reticulum. While here you can see that are present on the endoplasmic reticulum. So we can say that this dot are known as ribosome. So if in a endoplasmic reticulum ribosome are attached, then we called as a rough endoplasmic reticulum. If ribosome is absent, then we called as a smooth endoplasmic reticulum. Now you can see here, this picture is a Golgi apparatus. This is a ribosome. This is mitochondria. Here you can see the chloroplast, and chloroplast only present into the plant cell. These are the vacuoles, lysosome, and ribosome also attached with endoplasmic reticulum, as well as it freely present into the cytoplasma. So this is the 3D structure. For your exam point of view, I also saw you in a 2D structure. So once again we repeat: mitochondria, this is cell membrane. If a cell is if plant cell is present, this extra cell wall is present. In animal cell there is a no cell wall, as we discussed in previous episode. So here this is the only animal cell, so here only cell membrane is present. Inside the cell membrane there is a cytoplasma, centriole, this is centriole, or centrosome, chloroplast for the plant. Here it is a mitochondria, vacuoles, lysosome, ribosome. Small dot are ribosome here with the ER, endoplasmic reticulum, Golgi apparatus. So this is the 2D structure of the cell, and in which there is a cell organelles are present. So let's start with the first organelles that is present into the cell, and that is a nucleus. Nucleus is called as the control center. Why it's called as a control center? It is it perform all the tasks, it commanding center, commanding organelles. So we can say that the brain or boss of the cell. It stores the genetic material like a DNA. It directs all the cellular activities. It control. It regulate growth and reproduction, as well as responsible for heredity. It means it passes the gene from one generation to another generation. In simply language, we can say that nucleus is the boss, DNA is the instruction book, and organelles are the employee of the cell. So you can say that nucleus will decided all the what we have to do, and we how we can do, and that is the DNA provide the instruction book. According to that, all organelles are the doing their job. So or organelles are the employee of the cell. A similar example we can correlate with our college or university, or we can say in the school. There is a head is a present, or we can say principal is a present. So nucleus is a principal, and all faculty members doing their job, doing tasks like organelles. After nucleus, let's move to the endoplasmic reticulum. So we can say that endoplasmic reticulum have two types of endoplasmic reticulum: rough and smooth endoplasmic reticulum. As we already discussed, if on ER there is a dot are present, and that dot are we called as a ribosome. If ribosome are present on the ER, then we called as a rough endoplasmic reticulum. If if ribosome is absent, then we called as a smooth endoplasmic reticulum. Both ER have a different function. For example, rough endoplasmic reticulum solely perform the task like a protein synthesis. It involved in a protein synthesis. So, site for protein synthesis and help in protein transportation. While in case of smooth endoplasmic reticulum, there is a no ribosome is present, and its main function is lipid and steroid synthesis, and it's also as well as may play a role in detoxification of drugs and as well as toxin. So this is all things about the rough endoplasmic reticulum and smooth endoplasmic reticulum. So first we discuss nucleus, control center of the cell. It From the nucleus there is a rough endoplasmic reticulum is attached. So we also discuss there protein synthesis will happen, how protein synthesis will happen. So whatever nucleus will give the command. So the rough endoplasmic reticulum synthesize the protein, and afterward next task comes that we will discuss later on. So this is the all about ER. Then we discuss about the ribosome. So ribosome is present, we already discussed, present on the endoplasmic reticulum, as well as it also present on in the cytoplasma freely. So protein factory of the cell, ribosome that will synthesize the protein. So we can say is the protein factory of the cell. It made up of ribosomal RNA and protein. So this is all about the ribosome. When we compare the ribosome between prokaryotic cell and eukaryotic cell, eukaryotic cell have a 80S ribosome, while in case of prokaryotic cell there is a 70S ribosome is present. So then it move towards this protein synthesis move from start from nucleus to er to Golgi body. So later on we are going to discuss about Golgi body. So for exam point of view, it may ask which organelles known as the site of protein synthesis, which organelles known is the site of protein synthesis. And for protein synthesis there is the one organelle, and that is ribosome. So in short, here we discuss about the three part: ER, both two kinds of ER, and ribosome. So then this is the same picture, endoplasmic reticulum and ribosome. So you can see that how this ribosome attach on ER. So you can see that one part ribosome having a two part, in 80S, 50 and 30S subunit are there, and these are the how they attach on the endoplasmic reticulum. Afterward, we are going to discuss about the Golgi body. So we can also set Golgi apparatus, and how they known as it the packaging and shipping center of the cell. So we already discussed from nucleus to rough endoplasmic reticulum. On the rough endoplasmic reticulum there is a ribosome is present, and this ribosome synthesize the protein. This synthesizing protein carry on the whatever rough ribosome prepare protein, and smooth prepare the lipid that will transport in the vesicle form into cis phase. So here you can see that this is the cis phase of the Golgi body, and along with this side phase that is known as trans phase. So from it this vesicle receive whatever protein synthesized, whatever lipid synthesized, that vesicle receive to side of the Golgi body. Afterward is shipping side. So from the trans phase is transport to the plasma membrane, protein and lipid. We all know plasma membrane are made up of lipid bilayer membrane. In between that, integral proteins are present. So membrane also required lipid as well as a protein. So that will that made by the endo of ribosome, and how several organelles are involved. Later on, in the hydrolytic enzyme are packaged into vesicle and sent to lysosome. So you can say we can say that this protein are transport into the lysosome that we will discuss in next slide. So here we can say that lysosome contain more than contain more than it contain dozen, sorry, it contain dozen of enzyme, and it perform the task perform the task. So these enzymes are made up of proteins, so it will transport into the lysosome, as well as some protein or cell also necessary outside of the cell. So it will secrete protein outside of the cell via exocytosis process. Exo, it means outside the cell, so it will transport into outside of the cell. So in short, we can say that Golgi, it perform protein and lipid are modification role. It's also sorting out at packaging and shipping whatever where it requires. So these are the function. So factory, when how we can compare this protein and lipid whatever synthesize. So analog with the factory, rough endoplasmic reticulum makes the protein. It transport to Golgi. Golgi will package and shipping center. Then vesicle, in the form of vesicle, it deliver into like a vehicle, and transport to what various destination like plasma membrane, lysosome, and it will secrete outside of the cell. So we can say that in the for exam point of view, what is the main function of Golgi apparatus? So it answer is modification, packaging, transport of protein as well as lipids. So move to the lysosome and vacuoles. This is the image of lyso. One side, left side, it is the lysosome. Lysosome, it means the cleanup crew. We can say that cleanup crew. It is a single membrane structure. It is a lipid bilayer surround the lysosome. It contain various digestive enzyme. We can say that it contain hydrolytic enzyme. Its role is a breakdown macromolecules. So it which uh, which cells are going to break down? So digest worn out organelles, old cell, food particles, various pathogenic microorganisms, that um, all are digest inside the lysosome structure. So how it will break down? So it whatever microorganism waste product that are enter into the lysosome structure, it this particle are fused with the lysozyme enzyme. This lysosome contain the lysozyme enzyme fused with then that will break down or digestion by the this various enzyme. If these are the recyclable, then it will useful nutrient are reused and waste are removed via exocytosis through. So for exam point of view, which organelles is known as suicidal bag of the cell? So this lysosome also called as a suicidal bag. So answer is lysosome. While in case of vacuoles are present in the both type of the cell, plant cell, animal cell. It is we also called as a storage unit. So in the vacuoles, these vacuole cell membrane that is called as a tonoplast. This is semi-permeable membrane that is surrounded by vacuoles. In between the in plant cell there is a large vacuoles are present in the plant cell, and while in case of animal there is a small vacuoles are present. But while in case of plant there is a it occupy a space to provide the turgidity, to maintain turgor pressure in plant. It What is the function of that vacuoles? So it store water, nutrient, pigment, as well as waste product. So this is the how types of vacuole in plant and animal. As already discussed, plant contain one large centrally vacuoles, while any animal many vacuoles are present, but in a very small size. In plant cell, it maintains turgor pressure, while here it temporary storage and transport of material. In plant cell, it keeps the cell rigid and healthy. So, in the plant cell, vacuoles can occupy up to how many percent? 90% of the cell volume. So here one important question you have to remember, in plant cell vacuoles occupy 90% of the cell volume. So move towards the peroxisome detox unit. What is the function of this peroxisome? And this is the structure of the peroxisome. This is small structure, single membrane bound organelles. It contains the oxidative enzyme, right? So what is the function of this peroxisome? It break down the fatty acid. How? So it break down very long chain fatty acid through the beta oxidation process. So this is the it involve in breakdown of the fat. We can say that fat or lipid. It similarly it also do the very important task, detoxification of harmful substance, and that is more important for our cell function. It neutralizes toxin and harmful byproduct like peroxide by using the catalase. It also contains the catalase enzyme. During our respiration system, our body can generate various byproduct like free radicals as well as hydrogen peroxide. If they are generated in a larger amount, then our body have a defense mechanism, and it will encounter that harmful product. If it a harmful product it not remove, then it will interact with our important molecules, important cell structure, and damage our cells. So this mechanism also parallelly important to remove harmful substances. So how it will occur? Harmful substances enter into the cell. Peroxisome use the enzyme like we already discussed. Catalase is break down H2O2 into water and oxygen. So it converted into harmless product. So mostly this peroxisome is present in animal cell, rarely or absent into the plant cell. Plant use glyoxisome instead of the peroxisome. So peroxisome mostly present into the animal cell. So did you know peroxisome contain the enzyme catalase which break down toxic hydrogen peroxide into which two kind of molecule? Water and hydrogen. Sorry. So this is all things about the peroxisome. Here now we are discuss about the glyoxisome. So we already discussed here mostly animal contain peroxisome, but plant contain glyoxisome. This is the difference between peroxisome and glyoxisome. We already discussed. Quickly we revise the peroxisome. Single membrane is present in that peroxisome. There is an important enzyme is that catalase, oxidase. So it will roll in detoxify the cell. It break down the various fat containing molecule by beta oxidation process. While in case of glyoxisome, it will found in the plant cell, especially it may be present, we already discussed, may be present or may not present. It is especially present in oil rich seed. For example, we can say groundnut, mustard seed, and it will so form during the germination. So this is the structure of glyoxisome. You can see that glyoxisome and peroxisome structure looks like a similar structure. Here also single membrane structure. Here also enzymes are present, while in glyoxisome, enzymes of glyoxylate cycle, it synthase isocitrate lyase as well as malate synthase. These are enzymes are present, which enzymes play role into glyoxylate cycle. So glyoxisome structure, in glyoxisome structure, it convert fat into sugar. So what happen in glyoxylate cycle? Fat convert into the sugar, right? So this is convert fat to sugar into the plant during the seed germination, and via we already discussed isocitrate lyase, malate synthase using this enzyme. It play very important into the glyoxylate cycle. So it may ask in a competitive exam which organelles involve glyoxylate cycle. So these are the glyoxisome involved uh, in the glyoxylate cycle. It also support the seedling growth. It provide energy, building blood for growing seed until it can photosynthesize. As we know, when we are doing seedling, meanwhile there is we cover with the soil. So during that there is a no photosynthesis process starts. Still in starting it will useful glyoxisome useful for germination. It provide the convert fat to sugar, so it make them alive cell until the photosynthesis process will start. So this whole things about the glyoxisome. So this is the whole things about today's lecture. So how you can remember all the organelles which are present into the cell? So here is a memory trick. Naughty rabbit eat green mangoes, chew leaves, visit cool park, and carry many walnuts. What does it means? N for nucleus, R means ribosome, endoplasmic reticulum, G for Golgi apparatus, and M for mitochondria. C means chloroplast, L for lysosome, V for vacuoles, C for centrosome, P for peroxisome, C for cytoskeleton, M for cell membrane, and W for cell wall. So these are the various organelles function. So nucleus we called as a boss of the cell. R means protein kitchen. It means protein factory. It generate it made the protein. Endoplasmic reticulum, we can say the cell highway. It will transport to the Golgi body. So Golgi body we can say is a delivery center. It involve in transportation. Mitochondria, that is powerhouse of the cell. Uh, Chloroplast, that is a solar panel. It provides the photosynthesis process. In lysosome we called as a suicide bag. It in Gujarati we called as a Atmaghatak Kothri, right? So this is the vacuole can store water. We can say that water tank is store the water, nutrient, so on. The centrosome involved in cell division. Peroxisome, it means detoxifier. We already discussed peroxisome, how it will detoxify. It contains various type of enzyme, so it can detoxify the cell. This is a cytoskeleton, cell skeleton. We can say that in a cell membrane security guard, which molecules go inside and which molecules go outside, that will decided by cell membrane. And this is the boundary wall, the cell wall. So this all the organelles, whether they are single membrane, there is a no membrane present, or double membrane security is present. So which organelles does not have a membrane, no membrane? So it is ribosome, nucleus, centrosome, and cytoskeleton. Double membrane structure organelles known as nucleus, mitochondria, and chloroplast. While in case of single membrane organelles, you have to remember endoplasmic reticulum, both kind, smooth as well as rough endoplasmic reticulum. With them, Golgi apparatus are involved. Lysosome, vacuoles, and peroxisome. All are the single membrane, double membrane, and no membranous organelles. So this is the trick how you remember no membrane organelles, real nice cell. Here is the shortcut. Double membrane organelles, you have to remember NMC. While single membrane organelles, every good learner value practice. So this is the whole things about the episode 5. This is the list of MCQ for episode 4. And these organelles, whatever we discuss, it also called as the endomembrane system and protein processing. So these are the list of MCQ question that you have to give me answer into the comment box. Thank you very much to this episode. Keep learning, keep exploring science. Thank you very much.